Okay, and to finish where I left off at, we're placing the pineapples into the oven. Uh, so the pineapples that with the 75% water activity, they'll sit in the oven for about 2 to 2.5 hours. The 50% water activity pineapples will sit from about 3 to 4 hours, and the ones set at 25% water activity will sit in the oven for 6 to 8 hours. Uh, and so for the again for the wildcrafted elderberries, these I already harvested these about 2 weeks ago, so they are already harvesting and hanging along their vines in my kitchen at room temperature, which is about 68 to 73 degrees. These will air dry for about 2 to 4 weeks, and then once they're dried, I'm going to place them in a glass mason jar, and I'll let them sit about 4 to 5 more days so that they can be able to absorb any condensation inside the jar. Afterwards, um, after both fruit fully dry, I'll then use a water activity meter, and I'll measure the water activity, which I can calculate with taking the vapor pressure exerted by the fruit sample and divide that by the pressure exerted by pure water in the closed system of the mason jars. Then the water activity meter will measure the water surrounding fruit divided by vapor pressure of pure water, resulting in a value range typically from anywhere from 0 to 1, 1 being 100% water activity and 0 being 0 water% activity. But for the most case, most microbes, you don't see any microbe activity at a water activity of maybe like 70%. So it'll be interesting to see what type of microbial growth they'll have in these fruit. After after the dehydration process of the fruit, all the samples will be placed inside the water activity cup, and then the machine will measure the water activity. The water activity will be recorded and the temperature, and then they'll be placed inside. I'll take notes of that, and I'll compare and evaluate that for my hypothesis to predict the shelf life of both of the fruit for the pineapples and the elderberries. And afterwards, I'll take more measurements and I'll calculate and record the average water activity in all the fruit that's set at 25%, 50%, 75, and 85%. And then I have a table with figure 1, and I'll be able to input those numbers in so that we can know exactly what the content of the water activity actually is. For data analysis, I also would like to evaluate microbial growth. So for this part of the experiment, we're going to take some of the microbial cultures that we find in the elderberries and the pineapples, and we're going to use agar plates, and we're going to take samples of the pineapples and the elderberries, and I'm going to use a sterile cotton swab to swab the fruit, and then I'll place it on the plate, close the plate off, and then I'll be able to put those plates inside of an incubator, or maybe just let them sit at room temperature for 20 to 48 hours and see how much microbial growth they have at 25%, 50%, 75%, and 85%. Same thing would apply to the elderberries, but the only difference is I won't have multiple samples of the elderberries. It'll just be one sample of elderberry, and I'll be able to swab that, streak that into the agar plate, and let those sit for 20 to 48 hours just to see how much microbial growth has occurred over that period of time. After I do all my swabs and I do all my streaks in my agar plates, I'll label all of the plates. I'll date them. I'll notate the water activity levels for each pineapple and elderberry, and also I'll incubate each of the plates at a room temperature ranging from anywhere from like 68 to 73 degrees for 20 to 48 hours. After the incubation period, the samples will be stained if I can have access to that type of equipment, because some bacteria you have to stain it to be able to see it, because some bacteria, they're like transparent under light, so the light can like just shine straight through them. So, you know, with dealing with microbes and microbiology, sometimes you have to stand the plates so that you can actually see the microbial growth under a binocular compound microscope, which, you know, we should have one in the microbiology lab. So in summary, based on evidence from all of the sources that I use, which is about 12 of them now, the effects of food water activity in dehydrated fruit can be lowered using different techniques to ensure the extension of shelf life in fruit. This can be achieved while also preserving the color, the bioactive compounds, the nutrients, and also minerals within a dehydrated fruit using these new technologies, such as edible coatings, post-harvest drip, and dehydration techniques using food dehydrators or conventional ovens. This here is my reference page, and any questions, feel free to ask any questions, and I'll be happy to answer any of your questions at the best of my ability. Thank you.